Next speaker is Manfred Laubichler. Manfred is a member of our center who does complex systems and developmental biology and history and literature and other things. Manfred.
Thanks, Randy. So when we decided what would be a good topic to further the conversations and advancing the mission of the center, which you can read here, improving human health by establishing evolutionary biology is an essential basic science for medicine. And then since it's ASU, it has to be worldwide. But the question that came up, evolutionary biology, what do we actually mean by evolutionary biology and what kind of evolutionary biology? Because it's a common feature in many areas where evolutionary biology gets applied to explain a variety of phenomena outside the traditional domain of evolution, that there's a tendency to ignore the last several decades of progress within evolutionary biology and apply some of the more simplistic models that have been popularized throughout the recent decades. So in that context, it became very clear that one of the interesting challenges if you apply evolutionary biology to medicine or to disease is that we have to figure out what does evolutionary biology have to say about the evolution of very complex phenotypes. And that often is not the subject of the more traditional models which operate on a certain sense of abstraction and simplification. So that was the whole context for setting up that workshop and getting going. And so what I'm trying to do is sort of take a step back from all the detailed talks that we have heard so far and try to emphasize some of the fundamental theoretical problems that basically came up in any one of them. And so that's the title here, because that's really the core essence of what does evolutionary biology have to say about the relationship between stability and vulnerability, or you phrase it in theoretical terms in evolutionary biology, between robustness and evolvability, because that's the ultimate trade-off. And in order to get at that, a merger of the perspectives from evolutionary theory and from complexity theory might give us some insights. So that's the basic idea. So an observation that basically is confirmed by everything that has been said before, that phenotypes, in particular also diseases, are recognized as complex systems. You didn't hear a single story where there is a very simple single cause that explains what's going on in the disease condition. It was all about a highly complex systems-based approach. Now, if you look into some theoretical advances within medicine, not necessarily evolutionary medicine yet, that has really been reflected in such principles such as network medicine. And so you could basically look at all the slides that you saw, all the complex networks and the consequences as an example of networks or network medicine or a network-based approach to human disease. And that's a review a couple of years old, and you can read it for yourself. It's basically we need to understand the multiple interdependencies and complex interactions between a whole range of different causal factors in those phenotypic systems. Barabasi, who's driving that from the point of view of complex systems theory, just came out with a book 3 days ago. And that's very interesting. Over 400 pages, 16 chapters, references to evolution, 0. So that tells you something about where a lot of the field is actually moving into. It is driven by big and diverse data sets, the ones that Ken and others were saying here. And that's a challenge. And the challenge is being met with a certain network and other complex systems concept-based approach. What's completely missing in all of those approaches is any reference to evolution. And literally, I have the book here if somebody wants to check. I got it 2 days ago. I looked through it. References to evolution, 0. Okay, so another observation based on the same starting point what went on within evolutionary theory. Now, in evolutionary theory, there's also an increasing move going away from the initial abstractions of genes, of alleles, of sort of single factors, of a simple population dynamics-based conception where you have variation within a population and then you throw some kind of dynamic calculus on it and then off you go and you explain why this gets selected and this not and all those kind of things. So we are moving beyond that. So give you a few examples about how that looks like. So this is a prime example that motivated some of those transformations in evolutionary biology. This is a slide of several fossils representing the Cambrian explosion. And what we are really interested in is how those diverse phenotypes, in that sense now high-order phylum-level phenotypes, could emerge in a very short period of time, and what are the underlying causal mechanisms, that means causal mechanisms of development that explain those different phenotypic features. So that's the field of developmental evolution. It brings in a complex systems perspective, now represented here through our understanding of how individuals develop in ontogeny, ruled and governed largely by complex underlying networks, as you will see in a moment, and exploring the evolutionary consequences of this. So this is how you can represent the causation in development that builds a complex phenotype. You start with a single cell and you have cell divisions and you see gradual differentiation into different cell types that then aggregate and interact to produce different phenotypes. And that process is highly complex, and of course a lot can go wrong here. From a point of view of evolution, of phenotypic evolution, whatever goes quote-unquote wrong here is what helps us explain patterns of phenotypic evolution, because those are certain types of variation. But of course not everything can vary, because anybody who has had any exposure to comparative biology, hopefully some of you in this room still, you will recognize that variation is highly discontinuous. Of all the millions and millions of animal species that exist and have existed, they fall into less than 50 major body plans. Why? So there's a lot of stability in there, plus there's a whole lot of variability or evolvability in the system. And this is where we're trying to get at this. Now, I have to also show a complex network. This is one that we work on. This is the sea urchin embryo. This basically describes the first 32 hours in the life of a sea urchin, if you're interested. Any particular changes in the structure of those networks help us to explain why certain features remain very stable and why other features show variability. And you see how we're slowly moving towards disease conditions or pathologies with that logic. You can analyze those networks, and we're doing this in a comparative fashion, and you recognize that there are certain modules, network motifs, that have particular topologies, both in terms of the network structure as well as in the phenotypic outcome. And there's a reasonable, manageable number of those that have been found so far. And those networks then become aggregates of those modules. So it's not that everything is connected to everything. And so this is the interesting challenge. So if you think about Fred's talk about those pathways, so there's a simplistic version, and then you add another dimension about the long-distance interactions. But the question is, are there any regularities if you would compare many of those pathways? And clearly the expectation is, given what we know about the way chemistry works, we might find some of those motifs on this more now higher level area. So this is sort of the challenge that we need to get at if you sort of try to turn this network and complex systems-based approach and fold it into an evolutionary system. You can then combine them and can explain whatever you want. That means whatever we have data for, skeletal differentiation, the sea urchin, pancreas, and so on. Okay, this is the interesting connection now to stability and evolvability. What has been noticed in analyzing those regulatory networks is that there are high-level motifs, regulatory motifs, where the structure, not the individual elements, that's the important point. You can basically exchange most of the genes in those networks. But if the structure of interactions is preserved, we see a certain hierarchy. And there is a recognition, as of course, this is science, and the sociology of science is very interesting. So Eric Davidson calls it kernels. Günther Wagner calls it character identity networks, basically the same thing. What it means is there are identifiable high-end regulatory motifs in those gene regulatory networks that are stable and that can be correlated with high-level characters such as phylum or superphylum level characters. And then you get more and more gradual diversification the more downstream you go into those networks. So in thinking about where those systems have potential vulnerabilities or where they can actually vary, we have to figure out where in those regulatory networks we're actually operating. Because there's a reason why certain things are highly conserved, and there might be two reasons. There's one that might be structural, but ultimately they are highly conserved because if you start messing there, game over, and so there's a very high selection pressure on the conservation of those. And that's the important thing, network structures, not individual genes. And I think this is something that we see in all of those talks here, that we need to figure out what is the appropriate unit of analysis for us to actually begin to understand the evolutionary dynamics of those systems. We can't just assume we are focusing on individual genes or sequences or whatever else we have been focusing so far. Now, this is another development that is relevant here. Particularly if you think back to Randy's talk, I think that comes up here a lot. That in a somewhat independent development within the history of evolutionary theory, we now, under the concept of niche construction, have some slightly different ideas about the many feedbacks between organisms and their fitness landscapes, to put it into the theoretical language of evolutionary biology. It's not that you have a fitness landscape out there and it sort of selects whatever happens in a variation you find in the population. The actions of those organisms actively construct that niche. And insofar as those constructed elements of a niche are stable, they basically fulfill the criteria of heritability. And so what you have here are multiple levels of inheritance systems. And this is particularly interesting in the case of the rise of obesity, the rise of psychopathologies, because we are, of course, living in a constructed niche right now where many factors that in a way interact with the regulatory motifs inside our bodies or minds have changed, but are stable enough that we basically as a consequence get a different evolutionary dynamics. And if you try to understand what are the systemic risks and perform an analysis, for us, it's not just the technology or the organism that we have to look at. We have to look at multiple layers of relevant context as a contributing factor to those systems. So what it means is we have to basically figure out where are the boundaries of those various systems. Okay. So that brings us then to, I think, a way how we can rephrase the evolutionary theory challenge here, the evolution of systemic risks. That means analyzing those systems in such a way that we begin to see where we actually get vulnerabilities, where we get change, and where we have stabilities, which in some cases also might be a problem because if they prevent adaptation to new challenges and to new environments. So I would call sort of, this is a slide that you all know because I stole it from Randy and he has used it several times. This is basically a framework of setting up the landscape of questions focusing on Tinbergen's 4 original questions, and that's really sort of drove a lot of evolutionary medicine. The question is, what do we need to add to this? Or how do we need to slightly rearrange that landscape to get at this question of the evolution of systemic risk and bringing in systematically perspectives from complexity theory. Okay, so what are the challenges? I already talked about it. So we have to analyze robustness and evolvability as complementary properties of emerging or evolving complex systems. So the challenge here is the following. I think if you go there further on. So there's evolutionary theory that needs to explain both stability and change. And this is where we have a problem. So I was pretty well behaved so far, so it has to change at some point in the talk. So of course evolutionary biologists know that stability exists and needs to be explained. But they're going about it in the same way as Ptolemaic astronomers went about trying to preserve basically a geocentric worldview. So stability gets explained by adding epicycles. It is not systematically derived from an understanding of the underlying substrate of evolving systems, because they basically look still at it, and our mathematics forces us to look at it largely through a set of objects with variational properties, and then we throw differential equations at it. Complexity theory is a different way. So we look at what are the structures and what do we know in a general term about those complex systems that we have seen in all the talks. And what are the evolutionary transformations of those systems? And if we begin to understand those, then we would be able to actually derive from within one theoretical framework both stability and change, and not just explaining stability with a few ad hoc hypotheses, so evolutionary storytelling. Okay, this is basically what I wanted to say here. And if we do that, then we actually would find also a systematic way of understanding what we mean by vulnerabilities in complex systems. Okay, the way we do this is, as an example, can be developmental evolution, as I told you this. So how do we do this? Now, I think the reason why I brought up network medicine, I think this is a very fascinating and important contribution, because the more we learn about the existence and the dimensionality, but also the unifying properties of those networks, the more clear it becomes, at least to some of us, that we can actually begin to see patterns and structures of transformations of those networks. And that's a different way of thinking about evolution. It's not, which variant has a higher fitness value. It's about which variant can actually exist based from a particular network structure into the next. It's more about the origin of variation than what happens to variation once it exists in a population. I think that's what we have to add. So that means we have to integrate those mechanistic approaches from developmental biology, physiology, and all those systems into the standard model of evolutionary dynamics. I'm not going into it, I don't have time, but that actually will help us to actually finally develop an adequate conception of history in evolutionary biology, which we don't have. And we can actually then extend those networks, because this is both the challenge and the beauty of networks, because the saying goes turtles all the way down. So networks can be extended. So you can, as we see, from a genomic network to an epigenetic network to a physiological network to a social network, and the mathematics is such that we can actually treat it as one object, better than if we just sort of have different ontological categories. So that's where the mathematical focus comes in. Okay, so we did some of that, and so concluding here, what we need is basically evolutionary network medicine 2.0. And let's see how far we get with this workshop to develop some ideas in that context.
It records it for our video.
Oh, I got it. Yeah. So, I mean, this is a kind of a peripheral question. When you were, is this on? Yeah. Okay. When you were showing the development of the sea urchin embryo over 36 hours, so each of those different panels, were they representing sort of a different window, snapshot window of that 36 hours, or was that 36 hours combined
36
in which a
hours
whole lot of
combined.
things, there's a whole lot of dynamics would have been lost in
Yes.
that.
So it's basically it's a compression of a whole time series, and the
Okay.
color coding behind basically then identifies regions defined as cell types and things like that.
And was that from Eric's book?
Yeah.
Okay. Thank you.
Questions? Others? Mary Jane? Could you ask
I was just thinking, so you think there's a need for principles of evolutionary transformations. Don't you think, do you object to, so you're calling complex traits, you're calling them networks. I just call them complex traits. And I think that is okay. I think we're talking about the same thing in different words. And the network conceptualization has been getting more and more refined, so it's useful. But I think that the principles of transformation that you would like to see have been outlined, because it's now clear that, or just, I think, but I don't know whether you agree, that what happens during evolution is reorganization at different levels of organization, reorganization of ancestral phenotypes. And there are a limited number of ways that happens, sometimes through hormonal manipulations of the phenotype in one sex, transformation to the other, heterochrony, heterotopy, and so forth. So I think those are principles, and they have to do with modular organization, which means breaking down things into networks. So I think this has sort of been approached anyway, and I wonder what you think is limited about that.
I think we completely agree that this is exactly what has been done in individual cases. So we have your own work and others, so you figure out how certain elements in the developmental system, in this sense, hormonal signaling or whatever it is, can actually mechanistically explain how you get from one phenotypic states to another. We can now collect all those data and actually represent them. It's a question of formal representation that I'm mostly raising here. Represent them as a transformation within the structure of such a complex network. Now, there are other cases where this is done. We now get, if you look at a lot of the literature about epigenetic regulation, the influence of basically post-transcriptional regulations on all of that. Where can you intervene in that system? So this gets us a whole natural history of possibilities. I'm a theorist by trade. So we are trying to figure out what conceptual and also formal framework can we come up with that actually could take all those individual cases that we are beginning to understand and make it a case of something general. And that's basically the call for here, because that's what we need to do in order to reach the same success that the atomistic gene-based abstraction got us for about several decades in evolutionary theory. Because right now we have a very good predictive mathematical theory in population and quantitative genetics, and we have a lot of individual cases, and you know the debates as well as I do in the field.
Yeah, no, I see.
Yeah, so I think we agree. For once, we agree.
Yeah, one final question, and then we'll move on to our next speaker.
So your last thought there,
Microphone up and breathe.
your last thought there was on the history of science or history. Is that where this goes now for you? Can you say it?
Well, it's a mixture of all what we do in science is always in a way connected to our interest in history also. But yeah, what I am saying here is that we have to, there's an advantage of having some very clearly defined formalizations, because we can show in the history of science that those formalizations actually bred the success of certain ideas. And to take advantage of the knowledge that we all have here about individual cases, I think we need to develop a theoretical framework that is adequate for those cases. And my argument is that the current framework of evolutionary theory, sort of the population genetic one, is not. So that's the call for what I was saying here.